And welcome to Ask the Expert. We are delighted to be having Per-Ola Carlsson. He's a professor of medical biology at Uppsala University in Sweden, joining us. He also serves as a senior consultant in endocrinology and diabetology at Uppsala University Hospital. And his research focus has during the years been on islet physiology, islet vascular biology, and vascular engraftment of transplanted pancreatic islets. The work includes basic science work on the microcirculation and oxygenation of pancreatic islets in correlation to islet function and metabolism, as well as the paracrine role of islet endothelial cells for beta cell function. Other studies have dealt with obstacles in engraftment of transplanted pancreatic islets and strategies to overcome these hurdles in order to improve results of pancreatic islet transplantation. I know EPITA here is planning a big conference on that coming up soon. In recent years, he's also focused on stem cell research, including cell therapy studies using mesenchymal stromal cells to prevent development of overt type 1 diabetes in newly diagnosed patients with type 1 diabetes, and on the use of oxygenated macrocapsules in clinical beta cell replacement and on clinical imaging of pancreatic cells by PET. He's authored more than 200 peer-reviewed publications. And a little bit about his company, NextCell Pharma AB, has developed a mesenchymal, sorry, I'm omitting all these people as they're joining. There's a lot of people coming. NextCell Pharma AB has developed a mesenchymal stromal cell therapy for the treatment of autoimmune disorders with T1D or type 1 diabetes as its principal interest. The company is situated in the south of Stockholm. It successfully led phase 1 and 2 trials in the treatment of type 1 diabetes with a vision to move towards a market authorization of its cell product after phase 3 confirmatory studies. So with that introduction, welcome. Thank you so much for taking the time out of your busy schedule. And yeah, I'd love to hear more about what's happening with NextCell Pharma.
Thank you, Monica, for your kind introduction. I could perhaps share screen then.
Yes.
So I will talk then on the subject of mesenchymal stromal cells to intervene in the development of type 1 diabetes. And just a quick background is that mesenchymal stromal cells, they are a cellular therapy, an ATMP, and what differs that a little bit from many other pharmaceuticals is they provide a possibility for a treatment effect via combined mechanism without the need of combining several drugs, because these kind of cells, like all cells, of course, produce a lot of different things and then can target several mechanisms. And we, of course, do not know which pathway is the most important one, but it will definitely have a multiple impact. That is perhaps of value in a disease such as type 1 diabetes. That is an heterogenic disease, and also there are a lot of things not known about the mechanism and the etiology of disease. I started a couple of years ago, actually, with mesenchymal stromal cells. And we started out back in 2010 doing interventions with mesenchymal stromal cells in adult patients newly diagnosed with type 1 diabetes. And we then selected an autologous treatment with bone marrow derived cells. And that paper was published back in 2015. I will just briefly go through it. And what we did was that we asked for informed consent of the subjects recently diagnosed with type 1 diabetes between 18 and 40 years of age. And then we included them in a study where we retrieved bone marrow from them, and to half of the recipients we gave bone marrow back in about 4 weeks when we had expanded the cells, the mesenchymal stromal cells, and we gave that as an intravenous infusion. And before introducing the cells, we did a mixed meal tolerance test, and we repeated the mixed meal tolerance test 1 year later. And then we obtained a preservation of the remaining insulin production, the remaining C-peptide production, to be honest, in the patients that were treated with mesenchymal stromal cells, that are the open bars, while the control group, which did not receive any cells, they received placebo, they decreased in C-peptide, as you can see, with a mean number of 15 to about 15%, and that was statistically significant for some comparisons, not with regard to area under the curve C-peptide percentage, but quite close. It was a small study, just 10 plus 10 patients. We had plans of continuing with this study into a larger study, but then we faced the ATMP requirements in Sweden. We changed needs of also an MPA approved study, and therefore we were approached actually by NextCell Pharma, which had a component on the market. And they did not have it on the market, but they have an ATMP product that they would like us to test, and that was based on mesenchymal stromal cells out of Protrans jelly, and it was pooled mesenchymal stromal cells. I don't have a relation to this company, that is a disclosure then, but I have only been the PI of these studies. And we did a study then that was quite close to the first study, in that we selected adult patients 18 to 14 years of age with a normal weight, 50 to 100 kilogram. They were otherwise healthy. They needed to have some C-peptide left, of course, and recently diagnosed, meaning in this case that they were within 2 years of diagnosis. And these individuals were then subjected to a first dose escalation study, followed by a blind placebo-controlled study. In the dose escalation study, we tested 3 different doses of mesenchymal stromal cells, namely 25,000 cells, followed by 100,000 cells, and lastly 200 million cells. I would say in the bone marrow-derived cell study, we did use all cells that we could retrieve, but it was in the mean range between 100 and 200 million cells. We had DSMB looking at these first 9 patients, and since everything looked nice, we were allowed to continue into then a blinded placebo-controlled study. It was a very small study too, only 10 plus 5 patients, and based on the DSMB evaluation and our own evaluation of the first 9 patients, we decided that we should treat with the highest dose than 200 million cells in the placebo-controlled part. We decided also that we would retreat the first 9 patients after 1 year with a second dose of cells, meaning that those that received 25 million cells, they received 25 million cells more after 1 year. The medium group, 100 million cells, and the high group, another 200 million cells. If we focus on this first part, the dose escalation part, we observed that after 1 year, that is after the first dose, 1 year after the first dose, we had not significantly lost any C-peptide production during the first year when comparing to mixed meal tolerance test, one before treatment and one year afterwards. And we both in the treatment dose of 100 million cells and the 200 million cells had, well, at least with the standard deviation, not obviously lost any insulin production. For the 200 million cell dose, the mean value was also very close to 100%. while the low dose had lost about 25% in mean during the first year. We then retreated, as mentioned, these 9 first patients and observed that 1 year after that, we could see that the 3 individuals treated with 200 million cells, 2 different doses, at 2 different occasions, they still had 100% of their original C-peptide production left, while those that had been retreated with 25 and 100 million cells, they seem to have lost during the second year. We have now followed all subjects up to at least 3 years of the study, and then we can see that those individuals that have been treated with 200 million cells at 2 occasions, and that is then 2 years after the last dose, they have a mean value of about 85%, but they still have an overlap to 100%, while we have only about 50% left of insulin production in the 25 million and 100 million dose. We will, of course, follow these subjects for another couple of years. The whole study is planned for 5 years, according to regulations, with new mixed meal tolerance test twice a year. For the blinded placebo-controlled study, we treated then 10 patients and had 5 placebo subjects. And after 1 year, it was turned into an observation period for another 4 years. And since we had an endpoint then after 1 year, not all of the individuals approved of continuing into the observation studies. We have lost a couple of individuals in the treatment arm, mainly due to the COVID pandemic, and that they did not want to travel over all of Sweden for follow-up. Actually, we recruited all patients from Sweden, but the country is rather large and they needed to go there many times by flight, so they did not want to come for follow-up in the observation study. Then we need to ask them again. If we look at the numbers, we can see that at the 1-year endpoint for the study, we could see that those individuals that were treated with Protrans, they had good preservation of their C-peptide production, about 90%, while those that were in the placebo arm, it was a larger variation, but then about 60–70% only left after 1 year. After 2 years, 2 years after inclusion, then we had still a difference between the Protrans treated and the placebo treated individuals, and the same was observed 3 years after treatment. There was a tendency to a continuous loss of C-peptide production also in the treated subjects, but they at all occasions had statistically significant better preservation of insulin production. At this stage, we have still 2 more years for them to be included in the study. And after the pandemic, actually, we are planning to recontact those individuals that were part of the MPA-approved first-year study, so that we could include for the 4th and 5-year follow-up also all, hopefully all of the 10 subjects included in the original study, and not only the 6 presently followed. If we look at this, we can of course understand that we have lost insulin production over time. This is the micrograph that more or less shows how insulin production is lost during type 1 diabetes. And then hopefully we have a treatment that can preserve and decrease the further loss of insulin production for upcoming years, and then also delay any complications of disease. And if we look in the literature, this paper, for example, in Diabetes Care 2018, they report an exponential fall in insulin production over the first 7 years. If we calculate from that, and these are very small numbers, of course, but we can observe that patients that were given placebo in our study have less than 50% remaining insulin production after 1 to 1.5 years, while Protrans seem to delay this time to 50% remaining insulin to an expected 4.5 years. So if these numbers would be repeated in a larger study, we would have approximately a 3 years delay of insulin loss. I will conclude with some discussion on an ongoing trial we have. We don't have so much data on this one yet, but I would like to inform you that we also have a study ongoing in children and adolescents, and that is not sponsored by NextCell Pharma. It's sponsored by Uppsala University Hospital, so it's an investigator-initiated study. It's a double-blinded, randomized, parallel placebo-controlled trial where we test this treatment in individuals 7 to 21 years of age. And it started Q4 2021 with first patients included Q1 2022. It's a safety and efficacy trial then, and we include individuals with newly diagnosed type 1 diabetes within 6 months. And they should have remaining C-peptide then of at least 0.12 nanomole per liter. And we have this far performed the safety trial in the age group 12 to 18 years and also in the age group 7 to 11 years. And as you can see from this slide, there are in total then 6 individuals, 3 + 3 individuals that have been treated during spring term 2022. And we had a 3-month follow-up of the subjects in September, last patient, 3 months after treatment. And then we had quite recently a DSMB that looked through the data, and they did not see any problems. We had no obvious adverse events relating to treatment. We have never seen that either in the adult subjects. So all in all, we have just been approved for continuing into phase 2 part here. And we will start screening next week for 30 patients in the age group 12 to 21 years. We will when we have included those and followed them, the last patient for at least 6 months, we will have a new DSMB and then open the trial for the age group 7 to 11 years, hopefully 30 individuals there too. And we aim to follow the subjects for a primary endpoint 1 year after treatment, but of course, in total 5-year follow-up observation period for safety and of course efficacy too. I would like to conclude. We have a research team that consisted of several MDs, specialists in both pediatrics or endocrinology and diabetology, and of course research nurses. We try to include subjects from all over Sweden, and in the children's study we hope to start up 2 new centers in other parts of Sweden too, hosted by Jonny Ludvigsson and Helena Elding Larsson, who will be co-PIs for the study. We also have, of course, some funding for these studies. I just received a grant from the Swedish Research Council for this pediatric study that will at least take some of the costs. Thank you for your attention.
Thank you very much. That was great. Really interesting work. And it's great to see the findings are really pointing in the right direction. I wanted to ask you a couple questions and then open it up to the larger group. I guess my first question is, what is the mechanism? What are you thinking about the mechanism for these results?
Well, of course, we don't know because we don't have a mechanistic studies at hand. There are, of course, experimental studies that have given some indications. I would say that we give the cells intravenously, and based on experimental studies, these cells will mainly then end up in the lungs, and they will exert a systemic immune modulatory effect. And based on experimental studies, they don't see so much homing. It could occur, but that has mainly been reported if the cells are administered arterially. And when comparing the studies with arterial and intravenous administrations, the results are as similar, I would say. So it's safer, and therefore we choose the intravenous route.
That's really interesting. How do you compare what you guys are seeing, what you're doing, to Paulo Freire's group at Enthera? Do you feel like they're parallel tracks or something very different?
All ATMPs, of course, differ to some degree. But it is, of course, very important that mesenchymal stromal cells from different sources can give similar results. I believe that one critical part is actually a low expansion of cells. Based on previous studies, mainly in the graft versus host disease field, they have been reporting much better survival of those treated subjects for that critical disease when the cells have been expanded only for 2 to 3 passages. And we try to adopt to that both in the bone marrow derived study and also in this study with water jelly derived cells. And the only way to do that mainly is to pool cells if we would like to have a repeatable product too. So the Protrans is a pooled product out of about 5 different donors, actually. So I think that's a critical component. I could add another thing that I did not mention in the trial. One of our endpoints for safety was also to look at HLA immunization, and we have not observed that, actually.
Okay, yeah, that's very interesting. And then I wondered if, what are your thoughts? I mean, do you have any hypotheses, I guess, around the idea that this could be one of, dual or triple therapies, right, in concert with other evolving therapies that are happening, that are being pursued right now? Thinking specifically of, the prevention study, group and the teplizumab studies, things like that. What are your thoughts? Could this be something that might be used in concert to either extend a prodrome or just kind of keep people in remission, quote unquote?
I believe that in any way, the study results still very small numbers, but based on the repeat studies, I would suggest that we need to retreat subjects with MSCs 1 or 2 years apart. I don't know about children, but based on the adult studies. If we should combine it with other treatments, it's difficult to say, actually. Of course, it could be tested, but I would like at first actually to test a study where we do this kind of repeated treatment, high dose, once yearly, to look at the long-term results, because it's not an obvious additive or synergistic effects with other drugs.
Mm-hmm. Yeah, I mean, so that's a big question. I know I threw that at you, sorry. I wanted to, there's somebody in the audience here, Cheryl Greenberg. Are there any monitoring of the MSCs over the long term? Are there any known bad effects that need to be monitored from those residual cells?
Actually, we have not seen any obvious side effects of treatment. We have, of course, looked for that from other fields where mainly graft versus host disease field, there have been indications of increased risk of infections, meaning upper respiratory tract infections and so on. And we of course looked at that. We have small number of patients, but it was not a statistically significant difference between the groups. But definitely we indeed plan to follow them for an extended period of time. Based on the graft versus host disease studies, and knowing that in that field several patients actually die irrespective if they receive treatment or not, they have done autopsy studies and they have not been able to retrieve actually treated subjects with allogeneic mesenchymal stromal cells. So it might be that these have some form of kiss and die effect, or whatever you call it, where they and it has been speculated that a lot of the effect is due to lysing of the cells intravascularly in the lung and cause a storm of cytokines and chemokines that can immune modulate. And so perhaps there are not so many cells remaining long term, actually.
We have a couple more questions, and please throw your questions in the chat or raise your hand and we can call on you. But here's Chloe Rackman. Great to see such encouraging data. My understanding is that MSCs are perhaps not as hypoimmunogenic as initially thought. What are your thoughts on MSC source, in particular the use of autologous or third party? Encouraging also to see the use of third party in more recent studies. Is it possible to comment on age of diagnosis?
Well, it's difficult to say if you should use autologous or allogeneic cells. I would say that we had a more or less similar effect of mesenchymal stromal cells from bone marrow or Wharton's jelly. A benefit, of course, with using Wharton's jelly derived cells is that you can establish a product, an ATMP. It's difficult to have an autologous production as an ATMP because you need to do several testing on each batch before release. And that makes it much simpler if you can pool cells and produce quite many cells that you can use as batches for many individuals, actually.
Okay.
And it will also give a repeatability, I would say, because if the cells are pooled from several different donors, there will not be so much difference between batches, because it could theoretically be differences between different donors with regard to efficacy.
We have a couple more questions. Do you have any future plans to use MSC-derived EVs for diabetes therapy? That's from Fantota Dejeuner.
I have a colleague at Uppsala University Hospital that works with manufacturing of such product. I don't have any plan presently to start such study, but if the occasion occurs, we will definitely look into that subject.
Who is that colleague?
Carl-Henrik Grinnemo. He's a thoracic surgeon.
Oh, fantastic. Another Reza Yarani, great talk. As an average, what was the weight of the individuals who participated in the study?
Sorry, I didn't hear that questions.
Oh, sorry. This was Reza Yarani. He said, great talk. As an average, what was the weight of the individuals that participated in the study?
Yeah, the span of the individual's weight between 50 and 100 kilos. I would say that most of the subjects had a BMI of about 25 or so. So for the females, it would have been about 60 and males about 70.
Okay. And then Yang Dai is asking, are younger recipients benefiting more than older ones, or is there any age difference?
We haven't obviously seen any effect in the small number of individuals tested in the adults. For the children, we don't have results of efficacy yet.
Okay, that's very interesting. Is there anyone else that'd like to have a question in the chat or ask something in person as part of the conversation? Feel free to raise your hand or unmute yourself. ask to unmute. I don't see anything here. Okay, one more question. Did this from Reza Irani, does this mean that the females received more cells?
Well, per kilogram, they reasonably did somewhat more, because we had a fixed dose between 50 and 100 kilograms. And that was for practical purposes, because the cells are frozen in bags of 100 million cells. So that we would have to if we had dosed it on per kilogram instead, we would needed to throw away cells. So it was some practical point of view that we think that the more cells, the merrier.
Someone has a question. People are really complimentary about the quality of the talk. But this Shashank is asking whether or not the MSCs are administered following fasting or after food. Does it matter at all?
I don't think it matters, actually. They were not fasting.
Mm-hmm. I guess also, it would be interesting to understand, there's a lot of talk about endotypes, in type 1 diabetes, and whether or not this type of treatment affects different groups differentially. I mean, it's a very, you have a small study, but I wondered if that's going to be factored into future studies.
Yeah, well, of course, we have genotyped everyone, and we do have some that have better preservation of C-peptide than others. I can't say that we have any difference with regard to genotype, so it could just be the aggressiveness of individual disease that differs, I would say. But of course, we will continue genotyping also in future studies to learn more.
Okay, well, this was fantastic, really interesting, and can't wait to see what comes up next. Also very interesting to see what might come of the EVs. That's a very hot topic right now. I guess in the interest of time and of everyone's time, I'd like to say thank you very much, as well as Taxa Mikit. That's what I've learned from. That's what I've learned from some of our Swedish supporters. So thank you again, Per-Ola. This was wonderful, and this will be available for listening and viewing on our website and our YouTube channel. Best of luck, and we'll talk with you again in the future, I hope soon. Bye-bye.
Thank you.